Hi there, myself Dr. Umesh Jagta, and today we are going to deal with the spectroscopy techniques. Before going to start to the spectroscopy technique, let's begin with the question: what is the spectroscopy? Spectroscopy is the study of the interaction of electromagnetic radiation in all its forms with the matter. The spectroscopy techniques is very important techniques in the biology, chemistry, forensic science, because in chemistry or in biology we have to carry out the different types of the reactions that are producing the some kind of the color, and that colored solution which is kept into the test tube. By looking at that the colored solution which is kept in the test tubes, we are uh, unable to say which type of the molecule is present in that test tube and how much of the quantity of that molecule is present, because we cannot measure that particular color. We only just if we carried out the reactions between the two chemicals A and B, and it produces the product C, and which is the colored product, that is some kind of say that is produces the yellow color. But while looking at just towards the color, we are unable to see which type of the molecule is present and how much of the quantity of that particular molecule is in that particular solution. So here we can unable to say or we can unable to give that particular information to the people. And here's the comes that is a spectroscopy. So the spectroscopy is the technique which includes the study of the interaction of electromagnetic radiations with all its forms with the matter, how that particular electromagnetic radiations reacts with the matter. And these interactions of the electromagnetic radiations, we know that is the electromagnetic radiations, or we call as the electromagnetic spectrum. It consists of the different types of the rays, that is ultraviolet radiations, infrared radiations, uh, x-rays, gamma rays, and every one of these particular kind of the electromagnetic radiations are interacts with the matter in a specific way. And we can use such interactions to find out the information related to the certain kinds of the substances. And each one of these electromagnetic radiations reacts with the specific uh, matter, and it gives the some kind of the information. So these interactions might be that is a electronic excitations. Suppose if that is a UV, ultraviolet rays are interacts with the matter, it produces that is the electronic excitations, or that particular uh, molecular vibrations, if that is the IR, infrared radiations, are interacts with the matter, it produces the molecular vibrations like that of the there is a NMR, uh, that is a nuclear magnetic resonance. And in this particular nuclear magnetic resonance, we use the different kinds of the rays. So each type of the electromagnetic radiations interacts with the matter in a specific way. So you remember that is the spectroscopy is nothing but the study of the interactions of the electromagnetic radiations with the matter. So this is a very popular experiment you carried out in your school days. That is the if a beam of a white light is passing through a prism, it is separated into the various kinds of the components. Okay, so if a white light is passing through this prism, it produces the different types of the various components, and these are the seven colors you called as the that is the Roy G blue, means R for red, O for orange, Y for yellow, G for green, B for blue, I for indigo, and V for violet. So that is our normal light is consist of and the different types of and the colors. And we see in the case of that is the rainbow. So this white light is splits into the several types of the colors. And these optical systems which allows production and viewing of the spectrum, it is called as a spectroscope. So how we can study these interactions of the electromagnetic radiations with the matter? So we need a specific type of the instruments, and these optical system or the instruments which allows the production and viewing of the spectrum, it is called as a spectroscope. There are many other forms of light which are not visible to the human eye, and spectroscopy is extended to cover all these. Okay, so human eye, you know that is a human eye only sees that is a visible range, that is a white light, and which produces the different seven types of and the colors. Why we are seeing that is the colors, why we see that is a blackboard is white or particular yellow, or our plant is the green in color, because we are not going to see the absorbed light. So white light, when white light strikes to the any object, it absorbs the sum of the colors or the sum of the wavelengths and reflects the one of these. So suppose why the plants are look like a green, because plants absorbs the wavelength, uh, and it only reflects that is a green. So that particular plant appears to green in color. Why blackboards is looking black? Because the blackboards is a black in color, it absorbs all the colors, so it's gives that is a black in appearance. White color, so white color reflects all of the radiation, so it constitutes as a appears as a white. Okay, so we are a C, that is a only C, that is a reflected colors. And apart from this visible light, we are unable to see other forms of light. And to see that is the other forms of light besides the visible light, we need that is a spectroscopy techniques. So in absorption spectroscopy, we measure the amount of light absorbed as a function of wavelength. Absorption measurement can give both qualitative and quantitative information about the sample. So absorption spectroscopy is not only for the qualitative information, but it also give the quantitative information, that we can measure how much of the quantity is present in that particular solution. Absorption spectroscopy in the UV, that is ultraviolet visible region of the spectrum, because it is so widely used in the chemistry, biology, forensic science, engineering, agriculture, clinical chemistry, and many other fields. So we going to move, that is the next, that is the absorption process. Every molecular species is capable of absorbing its own characteristic frequencies of electromagnetic radiation. This is very important. Every molecular species is capable of absorbing its own characteristic frequencies of electromagnetic radiation. So this is the first type of the absorption spectroscopy, and we are going to see that is the principle and applications of the ultraviolet visible spectroscopy, that is the UV visible spectroscopy. So you know that is on the electromagnetic radiations. UV radiations are lies starts from wavelength range from 220 to 380 nanometer, and visible radiations is the 380 to 780 nanometer. Substances can absorb varying amounts of UV, ultraviolet, and or visible radiations at particular wavelengths. Colored compound absorbs energy in both UV and visible region of the electromagnetic spectrum, and that particular substances which can be liquids and the solids, and measurements are made with instruments called as the spectrophotometers or simply spectrometer. Modern instruments can be coupled to the microscopes which allow solid samples and very small samples of solids and liquids to be analyzed both qualitatively and the quantitatively. So, how that is particular UV visible spectroscopy works. So, if a particular wavelength of ultraviolet or visible radiations can be isolated from the source and passed through the sample. Okay, so this is the sample, and we are going to strike a particular wavelength light on that particular transfer. So when this light having the intensity I0, when this light is passing through this sample, the sum of the light or the sum of the radiations are absorbed by the compound or the sample which is present into the cuvette, and some of the light is transmitted from this sample. So this is the transmitted light, and which is denoted by the IT, that is the transmittance intensity, will less than the incident light I0. Okay, you understand? Suppose if we strike this sample a particular light or having the particular wavelength and the intensity, and this intensity I0, suppose here the 100% light is striking to that substance, while this light is passing through the sample, some of the light is absorbed by the sample and the rest of the light is transmitted or passed through this sample, and this intensity of that light is given into the IT. So the amount of light transmitted with respect to the incident light, amount of light transmitted with respect to the incident light is called as a transmittance, and that transmittance T is the ratio of amount of light or the intensity of the light transmitted divided by the uh, intensity of light I0. Okay, so I0 is the incident light intensity and IT is the transmitted light intensity. So sample can absorbs all or the none of the incident light, but this sample is absorbs all. Suppose that is 100% light, so your sample going to absorb all the 100% light uh, or none of the light, and therefore transmittance often quoted as a percentage. So percentage T, percentage transmittance, is equal to transmitted light intensity divided by incident light intensity multiplied by 100. Uh, what are the laws of the spectrophotometry? You are aware about there are the two very important and basic laws regarding with the spectrophotometry, and third law, it is a combination of the first two laws. The first law is given by the Lambert, and it is called as a Lambert's law. According to the Lambert's law, absorbance, which is denoted by the capital A, is proportional to the path length L of the absorbing medium. According to the Lambert's law, absorbance A is the proportional to the path length L of the absorbing medium. And the Beer's law states that absorbance A is proportional to the concentration of the sample, that is the denoted by the C. So Lambert's law is telling that that is the absorbance is proportional to the path length of the absorbing medium. That is a path length means uh, path length is the length of the cuvette in which we can place the sample. Okay, and this length is having the different sizes. So suppose that is 0.5 centimeter or the 1 centimeter, but all of the spectrophotometric cuvettes are comes into the 1 centimeter into the length. So that is a path length. And Beer's law states that absorbance is directly proportional to the concentration of the sample. that is the how much of the if concentration is present in that particular sample. So absorbance is present in the concentration. As concentration goes on increasing, the absorbance is goes on increasing. So these two laws are derived independently by the Lamberts and the Peirce. So Lambert states that absorbance is directly proportional to the path length. Peirce law states that the absorbance is proportional to the concentration of the sample, that is a C. Then these two law combines together, and the third law is derived, that is called as a Beer-Lambert law. And according to the Beer-Lambert law, the absorbance A is proportional to the concentration of the sample multiplied by the path length of the absorbing medium. So absorbance is directly proportional to the concentration of the sample as well as the path length of the absorbing medium, that is the L. And A is equal to epsilon C into L, that is epsilon is the molar extinction coefficient. So if you know that is a molar extinction coefficient, you can directly calculate that is a concentration of that the sample. You know the absorbance, you know the molar extinction coefficient of that particular sample, and path length. You can go into the calculate that is the actual concentration of the substance. So absorbance, it is the ratio, and therefore that particular absorbance having has no units. So this diagram showing the arrangement of the UV visible spectrophotometer. So UV visible spectrophotometer, actually this is the single beam UV visible spectrophotometer. It consists of and the source. After the source there is a filter, or it's called as monochromator. Then there is a shutter, then the reference cell, and there is a sample cell. So reference cell, it is the sample holding cuvette. In the reference cell we can put that is the standard compound, and in the sample cell we can put that is our testing sample. Okay, then there is a photo detector, amplifier, and the readout, or that is the display. So we go step by step from by each of these components. So UV visible spectrophotometer, that is the first comes, that is the source, and this light source is must be there, because the source is generate a beam of radiation that is sufficiently powerful for easy detection and the measurement. So this is the light source. Why we are needing the light source? To generate the beam of the radiation, and its output power should be stable for reasonable periods of the time. So source, light source is very important. It generates the radiation, and this particular radiation is must be stable for reasonable periods of the time. And there are the different spectroscopic sources, and these are the two types. One are called as a continuum light sources. So continuum, that is the continuous, the light sources which emits the radiations that changes intensity only slowly as a function of the wavelength. So these are called as a continuum sources. So in this figure, this blue line indicates that is the continuum sources, how that particular intensity slowly changes over the wavelengths. And another spectroscopic light sources, it is called as a line sources. So line sources which emits the limited number of spectral lines, and each of which spans a very narrow wavelength range. So in this is the B figure, B spectrograph, you can see that is the line sources. So line sources each produce a limited number of spectral lines, and which spans over a narrow wavelength range. Okay, so there are the two spectroscopic sources, two types of the spectroscopic sources. One, it is a continuum sources that produces the continuous uh, that emits the continuous radiations that changes intensity only slowly as function of wavelength. So they are not going to change their wavelength as fastly as compared to the line sources. And line sources which emits the limited number of the radiations of spectral lines which spans over the narrow range. Uh, Sources can be also classified as in continuous sources and the pulsar sources. So continuous sources again continue, and this is the continuous sources which refer the fact that they emit radiation continuously. If that particular light source emits radiations continuously, it's called as a continuous sources. And pulsar sources which emits the radiation in bursts. They produce the radiations, but they produce the radiations at particular bursts. They are not going to give the radiations in the continuous form. So these are the sources which are used for to the optical spectroscopy. So this is a helium and deuterium lamps which are produces the wavelength region that is 160 to the 380 nanometers, and these are used for the UV molecular absorption. Tungsten halogen lamp which produces the wavelength region of that from 240 nanometer to 2500 nanometers, and these are actually used into the UV visible near infrared molecular absorption spectroscopy. Then there is a tungsten lamp is also used for to the generation as a continuum sources for the optical spectroscopy. So, in UV visible spectrophotometer, a tungsten filament lamp which provides the broad distribution of wavelengths from 320 nanometer to the 2500 nanometers. These tungsten filaments are generally used. And these tungsten halogen lamps, also called as the quartz halogen lamps, that contains a small amount of iodine within the quartz envelope that houses the filaments. So quartz allows the filament to be operated at the temperature of about 3500 Kelvin, leading to the higher intensities and extending the range of the lamp well into the UV. And one of the advantages is that the lifetime of the tungsten and halogen lamp is double than that of the ordinary tungsten filament lamp. Okay, so most of the UV visible spectrophotometer that is the recently they're having the use that is the tungsten halogen lamps, and some deuterium lamp also used in the most of the uh, spectrophotometer, but this deuterium lamp they are provides the radiations into the only that is a ultraviolet region. Then next that is the filter or monochromator, or we called as the wavelength selector. So we know that is the light source in UV visible. We can going to use that is tungsten halogen lamp as a light source, and this light source gives that is continuous radiations. And once this radiation produced, their next part component is a filter or the monochromator. So spectroscopic instruments into the ultraviolet and the visible regions are usually equipped with one or more devices to restrict the radiation being measured to narrow band and that is absorbed or emitted by the analyte. Such devices greatly enhance both the selectivity and the sensitivity of the instrument. So you know that that particular source is produces the uh, wavelengths or the lights region in different wavelengths starting from to the 380 to up to that is the uh, 200 2500 different wavelengths of the light, and we are required to select that is the light of that particular wavelength, and these wavelengths are we can select by using the filter or the monochromator. So in many instruments they use that is a monochromator or the filter to isolate the desired wavelength band so that only the band of interest is detected and the measured. Other use a spectrograph to spread out or disperse the wavelengths so that they can be detected with a multi-channel detector. So this is the monochromator, and it is called as a grating monochromator. So monochromators generally have a diffraction gratings. So what is the grating? So grating is an the glass slits on which there are having the ridges and furrows that are graved at the same distance, and these diffraction gratings disperse the radiation into its component wavelengths. So most gratings in the modern monochromators are the replica gratings, and which are obtained by the making castings of a master grating. So how that particular gratings are produced? So in the modern spectroscopy, most of the gratings are called as the replica gratings. So we can produce the replica from the original grating, that is called as a master grating, because if we go for develop or produce the gratings, but each every time that particular job is in the laborism, and we cannot be draw that is particular ridges and furrows at the same distance. Okay, so a grating for the ultraviolet and visible region typically has 50 to 6,000 grooves per mm. So how many grooves are there present in 1 mm? So in 1 mm there are the 50 to 6,000 grooves. So the construction of the good master grating is tedious, time consuming and expensive because the grooves must be identical in size, exactly parallel and equally paced over the length of the grating, that is the 3 to 10 centimeter. In older form of the spectroscopy, there are use the prism. They use the prism to disperse the light. Okay, so there are the prism monochromator, but now in advanced spectroscopy there are used that is a grating monochromators. Some of the use that is a filters. So what are the role of the filters? So filters are operated by blocking or absorbing all but a restricted band of radiations. And there are the two types of the filters used in the spectroscopy. One, it is called as the interference filters and the absorption filters. And the interference filters are typically used for the absorption measurements, and these filters generally transfers a much greater fraction of radiation at their nominal wavelengths than do absorption filters. So interference filters are used with the ultraviolet and visible radiations as well as with wavelengths as long as 14 mm in the infrared region. So this is the graph, that is the percent transmittance versus the wavelength nanometer, and there is a C, that is a bandwidth for the two types of the filters. This is the absorption filter and this is the interference filter. So for the interference filter bandwidth it is the 10 nanometer, and for the absorption filter the bandwidth it is approximately 50 nanometer. The next component in the UV visible spectrophotometer, it is the reference cell holder or the sample cell holder. This is the single beam spectrophotometer, and you know that is the sample containers, which are usually called as the cells or the cuvettes, must have windows that are the transparent in the spectral region of the interest. Okay, so there are these reference cells or the sample cells in which we can put the sample. These samples container are called as the cells or the cuvettes, and these cuvettes are the square in shape, and on this square shape cells or the cuvette there is a two sides are they very transparent so that the rays are transmitted from these two sites. And these particular cuvettes are made of, that is, quartz or the fused silica. And the quartz or fused silica made cuvettes are required for the ultraviolet region, and may be used into the visible region and out of about 300 nanometer into the IR region. So the cuvettes are made up of the quartz and fused silica are used for the ultraviolet region, visible region, as well as the infrared region. And silicate glass is usually used for to the in the wavelengths range from 375 nanometer to 2000 nanometer region, because of its low cost as compared to the quartz and the next the quartz also is made up with the plastic cells are also used into the visible region. And that particular cuvettes are having the size of the 1 centimeter. Okay, so they are having the 1 centimeter into the size. And one care must be taken while we carried out the measuring, that is the absorption of that particular sample. That particular cuvette cells are free of fingerprints, grease, or the deposits on the walls may alter. Okay, so these cuvettes are very clean, because if you left the fingerprints on that surface of the cuvette or the grease or the deposit, you get the erroneous results. Okay, so these are the sample containers. So in this figure you see there is a various sizes of the different types of the cuvettes are in the shown, and typically these are only for the UV visible region. So this is a open top normal with lid. This is the stoppered cuvette. This is the stoppered semi micro cuvette. Okay, if you having the less sample volume, then you can go for this semi stoppered semi micro cuvette, then cylindrical, tall micro, minimum height micro sampling. So these are the various types of and the cuvettes. And you know that is the path length for this cuvette. It is the 1 centimeter. Okay, that is path length, means that is the breadth of the or the length of this cuvette. Next component, it is called as the photo detector. And what are the photo detectors? So photo means light and detector. So those detectors which are used or for the detection of the intensity of the light, it is called as a photo detector. And this detector detects the lights which are transmitted from these particularly cuvettes or the sample. And so this transmitted light signal is at the very low intensity, so that the amplifier unable to read that particular detector or display, they having the uh, cannot detect such a low intensity of the light. So this light intensity signal is multiplied into the larger quantity, so it can be able to detect. So that's why we need the amplifier. Okay, so the purpose of the amplifier is to amplify the signals many times, so we can get the clear and the recordable signals. This is the particular single beam. There are the two types of the UV visible spectrophotometer. One, it is called as a single beam. Why it is called as single beam? So single beam instruments are only suited for the quantitative absorption measurement at a single wavelength. Single beams means the single wavelength, and in which there is and the only one sample holder or the cuvette holder's place is there. So we can make one by one reading. So first we can take a reading of the standard or the blank, and after that we can remove that particular cuvette, wash it, and we can again fill with the sample, and then we can measure the absorbance of that particular sample. So we cannot be in single beam, we cannot be measure, that is the absorbance of a particular standard and the sample. Okay, so we particular firstly we can measure the reference then or the standard, and then we can remove that cuvette, and again the same holder we can place the sample cuvette that holding the sample. Then there is the double beam. So in the double beam spectrophotometer, in this design, the beams are get separated in time by rotating sector. So here, suppose this is a light source. This produces the emitting light radiations, and these are passing with the monochromator. So for which we can be place here that is a beam splitter. So once a particular wavelength is fall onto this beam splitter, these are splits into the two beams. So one beam is directly passing to the reference cell in which we can put our the standard standard compound against which we can read the sample, and another there are the sample cell we can put the sample cell. So in single beam only one cuvette holder is there, and we can measure by removing one sample after the another sample. But here we can directly measure the two samples, one with the reference and another with the sample, because the double beam spectrophotometer it consists of and the two cuvette holders. One in which in one cuvette we can put the reference or the standard substance, and in the another we can put that is a sample set. And after this, the one beam is passing to the reference cell, another beam is passing to the sample cell, and the intensity of is detected by the photodetector, and this photodetector detects the difference between the two signal. One is coming from the reference cell and another is coming from the sample cell, and later on this algorithm is used and they're showing the readout. So this double beam spectrophotometer offers the advantage that they they gives you the direct continuous recording and quantitative recording of that particular sample. So we have seen that is the different components of the ultraviolet visible spectrophotometer. And there are the two types of ultraviolet visible spectrophotometer. One it is called as the single beam spectrophotometer, and another it is called as a double beam spectrophotometer. Now, where we can going to use that is a UV visible spectrophotometer, and what are the applications of this UV visible spectrophotometer. So UV visible spectrophotometer is used to identify and determine many inorganic, organic, and the biochemical components. Uh, Ultraviolet and visible absorption spectrophotometer is used for the quantitative analysis, and is probably applied more extensively in the chemical and the clinical laboratories for any other single technique. So this is a very widely use techniques for the qualitative and the quantitative measurements of and that particular components in the field of that is the chemistry, biology, forensic sciences. So for example, here that particular visible spectrophotometer is used for study the different phytochemicals present into the plants. These techniques are used for the qualitative and the quantitative measurements of and the nucleic acids. uh, Okay, so these are the different uses of the UV visible spectrophotometer, and uh, you can check the different books and note down the various applications of the spectrophotometer in the field of biology, chemistry, or the plant sciences. Okay, so this is the most widely and important technique into the analytical biochemistry. Thank you.